So, good afternoon and welcome back. I hope everybody found their way back to our second session for today. And I also hope you enjoyed the break and also that you checked out already our networking hub. So now I'm happy to introduce the next session with the title From Nanosafety to Nanomedicine, a 10-year perspective. The speaker of the session is Professor Bengt Fadel from the Karolinska Institute in Stockholm, Sweden. Professor Fadel will provide a perspective on nanosafety research conducted in the past decade and provide a view to the application of nanomaterials in medicine. He has been involved in several EU-funded and national projects focused on nanosafety, like for instance Marina, Biorima, but also the EU-funded graphene flagship. He will focus his talk on lessons learned from these projects with emphasis on the synthetic and biological identities of nanomaterials and also their interaction with biological systems. The lecture will take about 1 hour and is followed by a 30-minute Q&A session. So I kindly invite all participants to put their questions in the chat, also already during the talk. And then when the talk is finished, we will try to answer them and have a good discussion with you. So please make use of this opportunity and put your questions in the Q&A. So Bengt, the stage is yours.
Good afternoon. It's my pleasure to join this virtual training school and to have this opportunity to present a perspective on nanosafety and nanomedicine research. And as you can see, I'm calling in from Stockholm, Sweden this afternoon. I'd like to start off with one of our most downloaded, certainly not most cited, but one of our most popular publications. This is a study published in the British Medical Journal back in 2015, where we decided to cross-reference all of Bob Dylan's songs and lyrics with all of the papers available in PubMed at the time. And we discovered that there are more than 200 publications in which Bob Dylan's songs have been referenced in the title of the paper. And as you can see from this graphic, The Times They Are A-Changing is the most popular song. The question I'd like to raise in relation to nanotoxicology is whether the times are changing or are they still more or less the same. What do I mean by that? Well, nanotoxicology can be seen as a new scientific discipline, the science of engineered nanoparticles and their effects on human health and the environment. But it's important to recognize that nanotoxicology is also based on the foundation of classical particle and fiber toxicology, and that we can learn important lessons from classical toxicology. Take, for instance, the case of asbestos and the related pulmonary diseases. In my view, asbestos-induced diseases are really a result of the failure to apply already available information in order to prevent this occupational disease. And let me quote here from a paper published in The Lancet, where the authors write, "It is now recognized that the inhalation of dust generated in the manufacture of asbestos often gives rise to an insidious and progressive pulmonary fibrosis, which is distressing in its effects and disastrous in its consequences." And please note that this was published in 1930, so at this point we have known for more than a century of the hazardous effects of asbestos, and yet it seems that we are very slow to react and to respond and to protect our workers. Another example is silicosis, which is not a disease of the past. This is another pulmonary occupational disease. And there was a recent review or editorial on this topic in The Lancet called Deadly Denim. And this refers to the fact that silicosis has been known to occur in otherwise healthy and young individuals in the denim industry due to the practice of sandblasting of jeans, as you can see in this picture. So in fact, it is still very much a reality and This is just one example of the occupational exposure to hazardous particles and materials. Just to give one more example, this is information and images that I've borrowed from a review by Phil Democritou and colleagues at Harvard School of Public Health. And this refers to the case of metallic nanoparticles that are present in toner printers, so in the toner of commonplace laser printers. And the authors have been able to show the presence of the particles and the fact that they are emitted during the printing process. So just another example then to show that we are constantly exposed to particles, both nano-sized and micrometer-sized particles. This is another very nicely written paper. This is a review published in Annual Reviews of Public Health back in 2009 by André Nell and colleagues at the UCLA. The authors of this paper stated at the time that no human diseases have been ascribed to nanomaterials. But I think we have to qualify this statement perhaps to say that no human diseases are known to be ascribed to engineered nanomaterials, because we do know, and there's certainly plenty of evidence, epidemiological and other evidence, to support a link between particle exposure and human disease, including not only pulmonary disease, but also cardiovascular disease. And of course, the key question here is whether the nano-sized or micrometer-sized particles are to blame. Coming back again to asbestos and to the so-called pathogenic fiber paradigm, this is a diagram or schematic figure that I've borrowed from one of Ken Donaldson's review articles, and it illustrates the fact that a man-made fiber that is both long and thin and biopersistent is more likely to be pathogenic. And it's also perhaps important to note that this paradigm is chemically agnostic in the sense that the chemistry is not necessarily important here. What is important are, as I said, the physical properties, the length and the diameter or the aspect ratio of the material, and whether the material is biopersistent. Ken Donaldson's group published a classic paper in Nature Nanotechnology back in 2008, in which they showed that long and rigid multi-volt carbon nanotubes do in fact trigger inflammation in mice, and the effects were similar to those induced by asbestos fibers, and therefore the authors used the phrase asbestos-like pathogenicity in that paper. And I also cited another publication, an in vitro study at the bottom of this slide from Harri Alenius' group in Helsinki, again showing that long and rigid carbon nanotubes trigger inflammation in activated human macrophages in a manner similar to asbestos fibers, whereas short or tangled carbon nanotubes did not show the same type of toxicity. And we know from the literature review conducted by IARC, the International Agency for Research on Cancer, that some multi-volt carbon nanotubes actually should be considered as possibly carcinogenic to humans. In particular, the product or the nanotube known as multi-volt carbon nanotube 7. Whereas at the time, this was published in 2014, at the time the available evidence with regard to other multi-volt carbon nanotubes or single-volt carbon nanotubes was insufficient. But around the same time, or a few years later, the European Commission published a number of so-called nanodata landscape reports, where another group of carbon-based nanomaterials were in focus, namely graphene and graphene-related materials. And I quote from that study, from that report, this was published in 2017, based on the scarce available evidence, it cannot be excluded that some forms of graphene will be as potent a toxicant as carbon nanotubes. But I think that at least in my view, this is an oversimplification, because not all carbon nanotubes are alike, and certainly this is also true for graphene-based materials. Not graphene is in fact not one but several different types of materials, and so it may certainly be a prudent and cautious approach, but it seems to me an oversimplification to bundle all carbon-based materials into one group. And we recently discussed this in relation to carbon nanotubes in a commentary in Nature Nanotechnology, as you can see perhaps there at the bottom of the slide. This brings me to the so-called graphene flagship, which is a huge EU-funded project, a project running over 10 years. It was launched in 2013. The project is coordinated by Chalmers University of Technology in Sweden, but it includes partners from all across Europe, more than 150, I think now more than 170 academic and industry partners, as well as other associated partners. And importantly, within the graphene flagship, there is also a work package dealing with health and environmental effects of graphene-based materials. And the partners of this work package published a paper early on in the project. This was, as you can see here, this was published in Angewandte Chemie in 2014, in which a classification scheme was proposed for graphene-based materials. Basically, the suggestion is that one should focus on at least 3 different material properties that can be used to distinguish different graphene-based materials from one another. So, and the first property is the number of layers. So you can go from atomically thin graphene sheets to graphene, so-called graphene nanoplatelets, which are essentially almost chunks of graphite consisting of many, many layers of graphene. The second property is the carbon to oxygen ratio. So again, going from pure graphene to graphene oxide, which is essentially 50% carbon and 50% oxygen. And the third parameter illustrated here is the average lateral dimension. So a graphene sheet can be either nano in lateral dimensions or micrometer sized or bigger. And we know from research conducted in the graphene flagship that these different material properties certainly dictate the toxicity or biocompatibility of this group of materials. And now, is this really important, or is it more of an academic question? Actually, I think it is very important and very relevant. And just to illustrate the point, this is an illustration of this issue. So very recently, actually in April of this year, Health Canada, the regulatory agency in Canada, issued a recall or a ban essentially on face masks containing graphene. We all know that there are plenty of different types of face masks on the market, but Health Canada zoomed in on this one particular product, which is actually a face mask with multiple layers and in which one of the layers contains graphene, or one type of graphene. And they the literature that they refer to mainly concerns graphene nanoplatelets and pulmonary effects of graphene nanoplatelets in animals. And it is known that graphene nanoplatelets may trigger pulmonary inflammation in mice. Is again, the question is whether this is a relevant comparison, but nevertheless, it tells us that it is important to understand the properties of the material, and it is certainly. I'm not going to defend the company in this case, because it's certainly up to each and every company who wants to put a new product on the market to prove it and to demonstrate its safety. But this, if you are interested in more research on this topic, I refer you to the review paper at the bottom of this slide, which is essentially a sort of midterm review of the work performed in the health and environment work package in the flagship. We published this paper about 5 years into the project. But the take-home message again is that we cannot a priori assume that all carbon-based nanomaterials behave in the same way. We know that short and tangled carbon nanotubes differ from long and rigid carbon nanotubes, and we know that graphene oxide behaves differently from graphene or reduced graphene oxide, and so forth and so on. Now, what else have we learned during this past 10 years or so in the field of nanotoxicology? I'd like to refer here to a report that was published in 2013. This was work coordinated by Kai Savolainen and a report jointly co-authored among the different members of the so-called EU NanoSafety cluster. And I just again quote from this report. We stated there that nanomaterials should be considered as biological entities and studied and perhaps also regulated as such. This was a strategic agenda type of report, and I think there are there's still a lot of interesting information to be found in this report, and you can download the report from the internet. But this particular point is something that I think deserves more attention. The fact that nanomaterials are not just chemicals, they're not just materials, they may actually interact and interface with biological systems in very particular ways, and we also discussed this in another review referenced at the bottom of this slide in annual reviews in pharmacology and toxicology. So again, an important lesson for all of us has been that nanomaterial characterization lies at the very heart of any nanotoxicological investigation. So in order to study the toxicity of a material, we have to know the material. And this is practically self-evident, but we have there have been great improvements in this regard in the nanotoxicology field. We discussed this in a review, as I mentioned here in this slide, a few years back, and the importance of characterization of nanoparticles both in nanotoxicology and nanomedicine. But I also refer you to this the second review here in Journal of Controlled Release, where the authors discuss the interesting fact, namely that those properties that we routinely characterize, morphology, surface charge, surface chemistry, are perhaps a good start, but additional and more precise parameters also need to be considered, because these additional parameters are really what determine the interaction of the particle with cells and tissues in the body. And some examples are illustrated here in the cartoon. And I think this is an important point. I mean, as George Whiteside said a few years ago, large things or particles or materials are what they are. But when things are small, they are what our measurements make them. So in other words, we are really dependent on the analytical tools and methods to tell us what the materials are and how they behave. And so this is a field that is also constantly under development, the actual analytical side of nanotoxicology. But in addition to knowing the material and its intrinsic properties, the physicochemical properties, we also now know that we have to understand the so-called biological identity, or which has been popularized as the biocorona concept. So in other words, when a particle is introduced into a biological system, it is very promptly coated with proteins and other biomolecules. And we refer to this as the protein corona. So I just would like to give you one example. This is from Warren Chan's group in Toronto. They published a paper in 2014 in which they studied, the authors of this paper studied 105 different gold nanoparticles, or gold nanoparticles with 105 different surface modifications. And they performed proteomics analysis and did so-called fingerprinting of the proteins that were absorbed to each and every one of these different nanoparticles. And those results are illustrated in the heat map in this slide. And then they looked at uptake of the particles using an A549 lung cancer cell line and determined and quantified the uptake by ICP-MS. And it turns out, as you may perhaps be able to see from the graph in the upper right corner, it turns out that this protein fingerprint is by far the best predictor of cellular uptake, and performs much better than other conventional parameters such as primary particle size measured by TM, or hydrodynamic diameter measured by DLS, or zeta potential, or the combination of all of the physicochemical properties. So the fingerprint, the protein corona, still outperforms the classical physicochemical properties in terms of predicting cellular uptake. So in other words, what does this mean? It means that the protein corona is also important. Naturally, the protein corona must, in one way or another, depend on the material properties. So there is an interplay, obviously, between the intrinsic material properties and the acquired properties that are governed by the protein corona. Just to follow on this topic, in nanomedicine in particular, we sometimes want to avoid cellular uptake. We want to endow the particles with stealthiness so that the particles can avoid macrophage uptake, for instance, and allow the particles to reach the target cells of interest. And this is an interesting paper, again from Nature Nanotechnology, from Katharina Landfester and colleagues. And these authors looked at naked particles versus particles functionalized with PEG or other polymers. Interestingly, they found that these polymers only provided stealth to the particles in the presence of plasma, so in the presence of a protein corona. As you can see from the confocal images to the right, you have one column in which cells are incubated in the absence of plasma, and to the far right, a column in which cells are incubated in the presence of plasma. So what this means is that you actually need to have PEG functionalization doesn't actually prevent protein adsorption altogether. Instead, you need to have some specific proteins adsorbed to the particles, and those proteins then act as repulsive proteins or dysopsonins, preventing the uptake of the particles. So it's a complex thing, the protein corona. It may sometimes promote uptake, it may sometimes prevent uptake by cells. And furthermore, this whole issue of PEG and pegylation of nanoparticles is perhaps not as simple or straightforward as it seems. Again, I already alluded to the fact that pegylation doesn't necessarily prevent protein adsorption altogether. It doesn't make particles invisible in that sense. But actually, when I was a student, these papers were published by other authors, but back in those days, clearly showing that yes, if you pegylate, if you have PEG molecules, and then the PEG molecules may provide repulsive forces at long range, as mentioned here, but at closer range, those interactions become attractive. So in other words, again, PEG doesn't automatically mean that particles can avoid interactions with proteins or cells. And so this, I think, is something that we need to revisit and to understand in much more detail if we are going to properly exploit polymer or PEG or other polymers for nanoparticles in medicine. Another key issue and an important one is the issue of translocation. And as I have pointed out in the title of this slide, I would argue that this might be one of the most critical features of a nanoparticle. So in other words, not the fact that the particle is small as such, but the fact that the small size of the particle allows it to translocate across biological barriers. And this wonderful illustration of the particle uptake following inhalation into the lung is taken from a review published in 2017 by Vicky Stone and many other experts in the field. And it illustrates the fact that translocation from the lung into systemic circulation might occur actually at least through two different mechanisms. And one is that the nanoparticles themselves, depending on their size, may translocate across the lung or air-blood barrier, so to speak. But the other possibility is that the particles are taken up by macrophages or other cells in the lung, and that these cells act as Trojan horses and bring the particles from the lung into either to lymph nodes or to other compartments in the body. And we discussed this also in a review published a few years ago together with Antonio Pietrojusti and Luisa Campagnolo, as you can see from the bottom of this slide. So corona formation is important, translocation is important, and I'd like to add a third parameter here, and that is biotransformation, or sometimes people might use the word degradation, but I think biotransformation is a better word. And this is an emerging understanding in the field of biomedicine and nanotoxicology, namely that nanoparticles may actually undergo changes, may be transformed in a biological system, in vivo, for instance, in tissues, in cells. And this is a very striking example published last year in PNAS, in which gold nanoparticles, which we typically believe to be very inert, biologically inert, gold nanoparticles in this case were examined over a period of many weeks and months, and were found to undergo biotransformation. So they were degraded or dissolved and then recrystallized to form new particles and so on. So I think, again, when we talk about characterization of nanoparticles, it's important to underline that we need to characterize particles as produced. That means when synthesized. But we also need to understand the behavior of the particles in the biological system, now meaning either in the body or in the environment for that matter. So this also applies, of course, to ecotoxicological studies. Just one more example. This was published in this month's issue of Nature Nanotechnology. This is a paper from Chunying Chen and colleagues at the Chinese Academy of Sciences. In this study, the authors investigated so-called molybdenum disulfide nanodots complexed with human serum albumin. So you have essentially a molybdenum-based 2D material complexed with albumin. And they administered these materials into the bloodstream of animals and then monitored the distribution and fate of the materials. And what you can see from this cartoon is that once the materials reach the liver, where you have your Kupffer cells and your hepatocytes, the materials were internalized and underwent biotransformation and giving essentially dissolution, giving rise to molybdate ions. And this is important because unlike many other metals, molybdenum is actually an essential trace element. So there are several molybdenum-dependent enzymes in the body, and what the authors argued in this study is that this excess amount of molybdate ions is somehow managed by the cells and leads to activation of those molybdenum-dependent enzymes in the liver. So again, I think a very relevant and interesting example of biotransformation, in this case of a metallic nanoparticle in the body. Now, the title of my talk somehow referenced the 10-year perspective on nanosafety, and I think it's appropriate to discuss here some of the lessons learned specifically from EU-funded projects within the nanosafety cluster. This diagram actually illustrates all of the nanosafety projects that were funded in FP7, and they are shown in red. And FP7 really, in many ways, was the golden era of nanosafety research, and as you can see from this diagram, and there were in fact 50 different nanosafety projects, but there were hundreds of unique partners involved in all of these projects. But among those partners, there were about 15 or so partner institutes that were represented across all the projects and somehow connected all the projects. And you can see these partner institutes in yellow. The JRC, the Joint Research Center of the European Commission, was not surprisingly involved in the largest number of projects among these partners, but you also have other well-known institutes like IOM, RIVM, EMPA, and so forth. Actually, I coordinated one of the first nanosafety projects in FP7, and we call this project NanoMune. And it was an interesting consortium consisting not only of European partners, but also US partners. You can see in this picture probably some familiar faces. Peter Wick, Harald Krug, and an uncharacteristically gloomy Lang Tran, somewhere there in the middle. Andrew Maynard, who was on our advisory board, and so on and so forth. And actually, the NanoMune project launched the training school in Venice 10 years ago. So that's how the training school started, with the strong support of Professor Antonio Marcomini, of course, from the Ca' Foscari University in Venice. And I'm very pleased to see that the training school has continued and thrived over the years. We also performed some very interesting research, and this is one example. This was a publication co-authored by US and EU partner institutes, and this is the first example of immune-mediated degradation of carbon nanotubes. So in this paper, we showed, together with Valerian Kagan and other colleagues, that human neutrophils contain or express an enzyme, myeloperoxidase, or MPO, which can digest single-walled carbon nanotubes under certain conditions. And we showed this, as you can see at the top of the slide, in the test tube, but also at the bottom of the slide in actual ex vivo activated human neutrophils. This is, I think, an important finding because it actually tells us that not all carbon nanotubes will actually align with the pathogenic fiber paradigm that I mentioned earlier, which dictates that or which tells us that materials that are biopersistent are more pathogenic. Clearly, certain types of carbon nanotubes can undergo degradation and are therefore not biopersistent in a biological system. And we have shown more recently that graphene oxide, for instance, can also undergo degradation, whereas graphene is much more difficult to digest through this mechanism. In many EU projects, we used nanomaterials that were provided to us by the nanomaterial repository at the JRC. And I think this is a very important resource and one that I would like to highlight here. Not only has the JRC provided aliquots of these different nanomaterials to multiple different academic labs across Europe, but they have also exhaustively characterized the materials as seen from these dossiers or reports that I have put in this slide. And so this allows us to compare results across different labs and between different projects for that matter. And I think that's very important. In one of these projects, actually the largest nanosafety projects to date, with some 85 partner institutes, the NANORIGO project, we were tasked with the screening of these materials. In the NANORIGO project, we focused on 19 different materials from the JRC nanomaterial repository. And we were tasked with the screening of these materials using a monocyte-like cell line called THP1. And we checked for cytotoxicity, but we also tested all of these materials for cytokine release at a non-toxic concentration of the material. And this is the result shown here after the multiplex cytokine array. It's difficult to distinguish any particular pattern here, but when we applied pathway analysis on the same data, and we then came up with this result, which is shown in the heat map on the left, which actually is pretty interesting because it shows us that we can group all of these 19 materials into only two groups, and namely those that activate nuclear receptor pathways and those that deactivate nuclear receptor pathways. And this is clearly seen here in the dendrogram at the top of the heat map, where you can see that there are two clusters that are formed. And those clusters are driven by PPAR signaling or LXR, RXR signaling. So in other words, even materials that are chemically distinct or different from one another may be grouped together or may show similar effects with respect to immune responses, and this may potentially be explained by an effect on nuclear receptor pathways in immune cells. Another project that has performed some very interesting work is the NanoMile project. And in NanoMile, one of the objectives was to establish high throughput screening, and the colleagues involved in this project focused on a set of 12 or 14 different metallic nanoparticles, as you can see from this slide, and managed to set up automated high throughput screening using a number of different cell lines and scoring the particles or the cells for a number of different cellular endpoints, as shown in this image. And so this is one way of speeding up the hazard assessment of nanomaterials, and certainly is something that can be applied to large numbers of particles. This was a smaller set of particles as a proof of principle, but a very powerful method. In another project, the NanoSolutions project, we studied 31 different nanomaterials that were synthesized by partners within the project, and the materials are listed at the top of this slide. So materials with different surface chemistry, different, I should say, core chemistry, and in addition, 4 different surface chemistries. So both metallic and carbon-based nanomaterials, but the materials were then varied with respect to their surface properties, either carboxylation or amino group functionalization or pegylation. This project was coordinated by Kai Savolainen, who is one of the foremost figures and leaders in the nanosafety field in Europe. And in this project, We not only synthesized materials, but we obviously tested the materials through a multi-tiered or multi-dimensional approach, as shown here, using various in vitro models, also in vivo models, different environmentally relevant species. And we used a number of different omics-based approaches. I'm not going to go into detail on the omics part today, but just to show you some of these screening studies performed in our lab, and this was published last year in Advanced Science. Here we use two different cell models. So again, the THP1 cell line, which is a model of human monocytes, but also primary human macrophages, so-called human monocyte-derived macrophages, HMDM. And this is just a snapshot of some of the data, but just to show you that for some particles, in this case titanium dioxide nanoparticles, we find that they are biologically inert or non-toxic at any rate, regardless of the surface functionalization. And you see those data to the left in this slide. But for some other particles, and the example given here are the silver nanoparticles, we see that the surface modification plays a significant role. So only the ammonium functionalized silver particles actually show a dose-dependent toxicity, whereas the carboxylated silver particles and the pegylated silver particles are apparently non-cytotoxic in these two cell models. The TM images in the middle of the slide there shows the uptake of titanium dioxide particles or clusters of particles, but despite the considerable uptake of particles, those nanoparticles are non-toxic for macrophages. We also performed cytokine profiling, again using the multiplex array to study 27, in this case 27 different cytokines and chemokines, and we could the results are illustrated here. So we do see to a varying extent the induction of pro or anti-inflammatory cytokines. We see also that the primary macrophages are more responsive than the THP1 cells, which is not surprising. What I haven't shown here, but what we have done is to apply the same pathway analysis as we did in the NANORIGO project. So we took all of this data illustrated in these two heat maps, and we performed pathway analysis, and we found again that we could group these 31 nanomaterials into two categories: the materials that activate nuclear receptors and materials that deactivate nuclear receptors. So this suggestion actually seems to hold true, which is that even when we test nanoparticles at a non-cytotoxic dose, we can still group the particles based on their inflammogenic potential using these in vitro models that I've illustrated here. The question that is often raised is how safe are nanomaterials? I mean, it's a question that we often get from journalists and from others, I think that we should be asking not how safe are nanomaterials, but how do we test nanomaterials? How do we test all the new and emerging materials on the nanoscale and the so-called advanced materials with respect to their safety? And I just took the Twitter page here from the Biorima project. Biorima is another very large project that focuses on this very topic, and as I've mentioned here, it's part of the Horizon 2020, and it's a project that is really devoted to the task of testing the tests with respect to human health and the environment. And the idea or the overall aim of Biorima is to develop a risk management framework for nanobiomaterials. I think this is perhaps something that will be discussed later on during the training school. When we refer here to nanobiomaterials, we refer to nanomaterials that are potentially useful or may be considered for nanomedicine applications. And I've illustrated some of those main types of applications in the cartoon here in this slide. And this is from an editorial published a few years back. The Biorima project was and is coordinated by Lang Tran, and I'm in charge of the toxicology work in the project. And this is one of the recent publications from the project. This was a study coordinated by colleagues in Venice, and it's not the final product as such. It's not the final risk management framework, but it's an outline of the risk management framework for nanobiomaterials envisioned by the Biorima project. And for more information on the project, please refer to the website shown there in the left-hand corner, biorima.eu. Just to give one example from the Biorima project, this is again work from our lab. In this case, we decided to use 3 different multi-walled carbon nanotubes, not because these materials are potentially useful in medicine, but because we wanted to test the tests. So we performed 3 different types of cell death assays, and I'm showing you here the results of the LDH release assay. Which in our hands seems to be a very robust assay for the monitoring of cytotoxicity of carbon nanotubes. What we find here is that the NM401, so-called NM401 material, which is the long and rigid nanotube, does in fact trigger cell death in a dose-dependent manner, whereas the tangled and sort of soft materials, non-rigid materials, NM400 and NM402, are apparently non-toxic. And we could show the same results using the Alamer Blue assay, which is based on a different principle than the LDH release assay. Again, showing that NM401 is the only cytotoxic material. When we apply the ATP content assay, we were unable really to draw any conclusions and due to interference with the assay. So it's important to apply the right assay. That's the message here, and because we know that some nanomaterials may interfere with some assays. We then went further, or my postdoc in this case went further and looked at the mechanism of cell death, and we found that the long and rigid multi-walled carbon nanotubes triggered lysosomal release of cathepsin B, and the results are shown to the left, and the confocal images simply show that cathepsin B is expressed in lysosomes in these cells, and the upper panel shows the release of cathepsin B only in the case of NM401, not in the case of the other carbon nanotubes. And this cathepsin B release sets into motion a cascade of events within the cell, resulting in inflammasome activation and caspase-1 activation, which leads to release of IL-1 beta from the cell. And we also found that this cell death is dependent on gasdermin D. So in other words, we were able to prove that the cell death in this case occurs through pyroptosis. And why is this important? Well, I think it's absolutely important to understand the mechanism of cytotoxicity of nanomaterials if we want to devise strategies to reduce or prevent the toxicity of those materials. In the last few minutes, maybe a few more words on nanomedicine. I think you are all familiar with the concept of the magic bullet and the idea of a targeted therapy, possibly a nanoparticle-based therapy that allows us to bring drugs to a specific target tissue and to avoid other tissues, and therefore to avoid unwanted side effects and toxicities. So this is essentially the promise of nanomedicine. But I refer you also to the review at the bottom of this slide, published almost 10 years ago in Science, in which the authors discuss the pros and cons of targeting of nanoparticles. It's a wonderful concept, but it's maybe not as easy and straightforward as it seems. One a successful example, however, is this paper published in Nature in 2010. This is a study of a small number of patients, but nonetheless human subjects, with a devastating type of cancer, skin cancer, and the authors of this study were able to design nanoparticles that were functionalized with a targeting molecule, in this case transferrin, on the surface, and particles that served as carriers of siRNA. So, and then they injected the particles into the patients and at some point extracted tumor tissue and were able to confirm ex vivo that the siRNA had the desired effect, which was to switch off a certain gene. So this was not a therapeutic study, but it was a sort of proof of principle study that RNA interference may actually work in vivo in humans. And this was possible thanks to the fact that they had used a targeted nanoparticle, which found its way to the cancer cells that express the transferrin receptor. But I have to say the nanomedicine field has been very much focused on cancer medicine and targeted therapy in cancer. But as we now know, the real breakthrough of nanomedicine is really in the area of vaccines. And one day these mRNA-based vaccines may also be used to treat cancer. But as of now, we certainly know that there are at least 2 mRNA-based nanoparticle vaccines that are being applied on a large scale against SARS-CoV-2. Actually, I have the mRNA-based vaccine, and I'm very happy for that, and I'm waiting for my second shot. So I hope you will not misunderstand what I'm trying to say here. I think this COVID-19 vaccines are fantastic. It shows actually, especially these mRNA-based vaccines, show really the power and the promise of nanomedicine. These vaccines are essentially lipid nanoparticles, so they're not liposomes, they're lipid nanoparticles containing mRNA, but they're pegylated particles, as illustrated here. And by the way, I put a number of references here, all excellent reviews on the topic of nanomedicine and COVID-19. The schematic figure here is from the most recent review by Andre Nell in ACS Nano. What are the potential drawbacks of these solid lipid nanoparticles and mRNA vaccines? Well, one is the fact that these vaccines rely on ultra cold storage, and so that is something that perhaps one can try to figure out, because perhaps by lyophilization or through some other redesign of the particle, because this cold storage chain is logistically very difficult to handle certainly in some parts of the world. Another possible drawback is the fact that there have been some very rare cases of strong adverse reactions towards these vaccines. Now, again, don't get me wrong. I think the vaccines are a fantastic thing, and I do look forward to the day when everyone has received their vaccination. But there have been some adverse cases, and the question is whether it's the lipid nanoparticle, is it the mRNA, or could it be the PEG moiety on the particle surface that is responsible for this rare adverse reaction? And that's something that I think we as a nanotoxicology and nanomedicine community need to look into. But the fact that we now have mRNA-based vaccines delivered with nanoparticles is really, I think, incredible. And again, as I said, something that may be potentially applied for therapeutic purposes in other diseases as well. We published a review on this topic last year in Nanomedicine. This was actually a sort of thesis project of Georgia Wilson-Jones, a very talented medical student from Rome, who was supposed to visit my lab in Stockholm, but then due to the pandemic was unable to do so. So instead we wrote a review and published the review together. And I think it was an interesting and fruitful exercise. And the schematic figure is taken from that review. So in the review, we hypothesized that it may be possible to create nano decoys, nanoparticles, which express the ACE2 receptor, and in order to intercept the virus so that the virus doesn't infect host cells expressing the ACE2 receptor. And interestingly, only a few months later, a paper was published in PNAS by other authors based on the very same concept. So, but not engineered nanoparticles. In fact, the authors took prepared cell-derived blebs or vesicles from THP1 cells expressing ACE2 on their surface, and they found that these particles or nanoblebs or nano decoys were in fact able to prevent SARS-CoV-2 infection in vitro, and also that these vesicles absorbed inflammatory cytokines, so they had a sort of dual mode of action. Then I think regrettably the authors claimed in the title of the paper that this nano decoy is protected against COVID-19, but that there's actually no evidence in the paper to suggest that this is true, because they used a mouse model of bacterial pneumonia, mice that had inhaled LPS. And so the nano decoys had some anti-inflammatory effect in that model, but I think it's still quite far from a model of actual COVID-19. Nevertheless, this is an interesting approach and one that I think could be developed further. We have also performed some COVID-19 related work in the Biorima project. I just want to show you one slide before we conclude. And here we tested silver nanoparticles without any functionalization or specific surface coating. And using a pseudovirus assay, as shown here, we were able to show that the silver nanoparticles could actually block infection of ACE2 expressing cells presumably by binding to the spike protein of the SARS-CoV-2. We also tested titanium dioxide nanoparticles. That's the green curve in the upper graph, and they had no effect or very little effect on the virus. If we look at the lower graph there, we compared the pre-infection scenario, which means that we first incubate particles with the virus, with the post-infection scenario, which means that we first infect the cells with the virus and then add the particles. And actually, as you can see, the pre-infection scenario is the one that works. So the particles are not having an effect on the cells. The particles are rather having an effect on the virus, and they're binding to the virus via the spike protein. I think with that, I'd like to wrap up and perhaps present you with some take-home messages. One is that we are constantly exposed to particles, both big and small, both micrometer-sized and nanoscale particles. And I just want to emphasize again that important lessons can be learned from toxicological studies of other man-made particles and fibers. I also tried to highlight a couple of different properties or features of nanoparticles that I think are quite important and that have emerged in recent years. One is the fact that, well, obviously we need to characterize particles, but we also need to understand that particles may undergo biotransformation in a biological system. So biotransformation is important and also presents a challenge. How do we characterize materials in situ in a biological system? biocorona formation or coronation is also important, because it's clear that the absorption of biomolecules, proteins and other molecules onto the surface of particles will impact on the particle interactions with cells and tissues. And I think the translocation of particles across barriers, across the cell membrane or across other barriers, the lung-blood barrier or the blood-brain barrier or the placenta barrier, this is also something that is fundamentally important and perhaps unique for nanoparticles. So I think overall there seems to be a blurring of boundaries, so to speak, between the intrinsic material properties and the biological sort of biological identity of nanomaterials. And this actually brings us back to the statement that we made already in the EU NanoSafety strategy document back in 2013, which is that nanomaterials are more than just chemicals. Nanomaterials should be considered as biological entities and studied and possibly regulated as such. I'd like to finish though with a proposal, and that this is a proposal taken from a recent book chapter that I wrote together with Phil Sayer, who is on the advisory board of the Biorima project. This is from a book called Handbook on Nanoethics, and the book is currently in press. And we refer to these guidelines as the Rest in Peace guidelines, the RIP guidelines for nanotoxicity testing. So I'd just like to run through this quickly before we close. The R stands for relevant. So we believe that toxicity tests should be relevant or realistic and relevant, and also generate results that are useful for risk assessment. The I stands for integrated or perhaps for intelligent testing. So integrated or tiered approaches where you start with an acellular model and then an in vitro model and eventually perhaps an in vivo model or an advanced alternative in vitro model and so on. So, but you don't screen all materials using animals. So an intelligent or tiered approach to testing. The P stands for predictive. So we need to make sure that our in vitro tests are predictive of actual relevant in vivo outcomes. But ideally, our test results should also allow for grouping of nanomaterials based on predictive testing of the nanomaterials. So these are the Rest in Peace guidelines. And finally, I'd like to thank the people involved in this work. First and foremost, all the postdocs who have worked in my lab in the various EU-funded projects, Nanomune, Marina, NANORIGO, Sun, Nanosolutions, and so on. And they are listed here. I hope I didn't forget anyone. Also, our US colleagues, in particular Valerian Kagan and Anna Shvedova, who played such an important role in the Nanomune project. Lang Tran, obviously, for coordinating both the Marina project and the Biorima project, two very important and very large projects that have produced, I think, a lot of important results. And Lang is also a very good friend. Also, the colleagues who were involved and played key roles in the Nanosolutions project, in particular Kai Savolainen and the project coordinator, and our many projects in the graphene flagship as listed here. And here are some of the members of the group, in particular Govind to the far right and Sandeep next to him, second from the right, who have been who generated some of the data that I showed you today. With that, thank you very much.
Great. So thank you very much, Bengt, for this very interesting and comprehensive talk. I enjoyed it very much. I'm sure it's the same for the other participants. And now it's time to move to the Q&A session. So we got a couple of questions. The first one from Samantha. You mentioned the gold ENPs were shown to undergo biotransformation in the Bulgaria et al. 2020 study, and so I was wondering what physicochemical changes were observed, and did this biotransformation affect the toxicological outcomes observed? Thank you.
Okay, yes, thanks Samantha for that question. That's actually an excellent question. Suzanne, my mic is on?
Yes, I hear you well.
Right, so that's an excellent question. So you're referring to the publication in PNAS on gold nanoparticles. It's not from our laboratory, but I can just give you a few brief comments. The authors used fibroblasts, and they managed to study gold particles in fibroblasts for a period of up to 6 months. How did they actually determine degradation? Well, they used various, I would say, high-resolution imaging approaches, high-resolution TM and SEM, for instance. And so it's very much a case of loss of structure of the materials. What they also found in that study actually is the recrystallization of materials that had once dissolved or been degraded. So you have a dynamic process whereby a particle first undergoes dissolution or degradation, but with time there may be a recrystallization and a formation of new particles, very tiny nanoparticles within the cells, and this was also shown in that study. As for the toxicity effects, I don't, to my recollection, I don't think they looked at this, it would also be problematic if the particles were applied at toxic doses, since they studied the cells for such a long period of time. Maybe one final comment, though. I mean, so I think your question is very appropriate, but one final comment is that these recrystallized particles that were observed in this particular model were, the authors suggested that they were sort of reminiscent of findings that have been observed in the past in patients who have received gold salts. As you may know, gold salts are used to treat rheumatoid arthritis. And one can certainly ask the question, where does the gold go? I mean, how does this gold therapy work, and what happens with the gold salts rather? But to some extent, they may also undergo recrystallization within cells and tissues. Does this lead to toxicity? Well, there are some cases in the medical literature of side effects of gold therapy, but again, I think you ask a very good question. So what happens when a nanomaterial undergoes biotransformation? Does it become more or less toxic? And the answer is that we don't really know, but that's a very good question.
Thank you very much. So another question is from Burkhard. Maybe a bit different topic, but do you see any significant toxicological potential from micro nanoplastics, or just a possible Trojan horse effect for other toxic substances? Thanks and best regards.
Okay, yes, thank you, Burkhard. That's also a very interesting topic, the topic of nano and microplastics. We have performed some studies in our laboratory on nano and microplastic, but I have to say that we use probably this particle that is used by many labs, which are the polystyrene nanoparticles and microparticles, which in our hands are non-toxic, at least in the cell models that we have looked at. But we have to qualify this statement because, first of all, and I think you are well aware of this, but first of all, these perfectly spherical and smooth particles that we are studying and others are studying are not necessarily a good model of the plastic debris that is found in the environment, which is probably of very heterogeneous shape and size. Secondly, which is I think you implied in your question, the plastic particles or debris could
act as carriers of other pollutants or chemicals, or that are either are somehow part of the plastic as such, the plasticizers and so on, or simply carriers of other pollutants. Plastic could also be carriers of pathogens, viruses, for instance, or bacteria. And perhaps, but this is maybe more on a hypothetical level, but plastic particles might acquire a corona in the environment. So let's call it an eco-corona, which might consist of allergens, for instance. So I think you are right, or you, as you imply, that perhaps particles, the main toxicity of this nano and micro-sized particles might be due to the Trojan horse effect, or the fact that the particles carry other pollutants into cells. But to fully answer that question, we have to also start to look at more realistic plastic debris or breakdown products of plastic, and not because I have a feeling that the literature is skewed towards these perfect polystyrene particles, which may, as I said, not be the real thing, really. So I hope that answers your question.
Thank you very much. There is another question from Volker. If nanomaterials are considered biological entities, where and how to draw the line to biological viruses, prions, and other such likes?
Okay. Yeah, thanks, Volker. That's quite a interesting and also challenging question. I suppose I have to blame myself for this, because I did say that nanoparticles are should be considered more like biological entities. And you are right. So where do we draw the line between an engineered nanoparticle with a biocorona of host-derived proteins versus a virus? Well, actually, I think they are, of course, they are not quite similar, but they are pretty similar, because a virus is also nano-sized. And a virus really is also somewhere in between a living and a dead object, because a virus is obviously carries its own nucleic acid and so on, but it's entirely dependent on the host cell for its replication. So it's somewhere between a living and a dead particle. Furthermore, viruses may also acquire a biocorona of host proteins. And this is something that was shown by other colleagues here at the Karolinska 1 or 2 years ago. And it makes sense. I mean, and it's not surprising, but it has not been studied much. So I think there are obvious differences, but also some similarities between nanoparticles and viruses. But the distinction I was also sort of trying to make, but it was maybe implied, is that nanoparticles are different from chemicals in the sense that they have a chemical composition. Yes, this is true, but they also have many other properties which, let's say, make particles different from the bulk chemical, and which allow particles to interact with biological systems in a way that chemicals may not. So that was sort of implied when I said that they should be considered as biological entities. But basically, the short answer to your question is that the difference is that they are still, at the end of the day, engineered and synthesized and not natural. But as I tried to explain now, I think there is a blurring of identities between all kinds of different man-made and natural and accidental particles. So that we can certainly learn lessons. I think virologists can learn lessons from toxicology, and I think that we as toxicologists can learn lessons from epidemiology and so on.
Great. Thank you very much for this answer to a not so easy question, I would say. I think these were all questions so far that were posted in the Q&A. So whoever is still online, feel free to put your questions in the chat. In the meantime, maybe I will ask you something, Bengt, if it's
Sure.
fine for you.
Yes.
So my question would be, in your opinion, where do you still see gaps in nanotoxicology research? So what do you think if you look ahead in the future? Now we talked about the past 10 years. If you think for the next decade, what do you think? How will nanosafety evolve, and what will be hot topic from your perspective?
Okay, that's a great question. Maybe George should answer, but I can maybe
Actually, maybe
make
if I can quickly comment, I think if I got George correctly, he somehow said that now after this more than a decade nanosafety research, we are more or less done with the topic. If I got his statement correctly,
Well,
I mean,
no,
would you
think,
agree, or do we still have gaps that we need to better address and that needs further
I think
research?
what I understood from the keynote lecture is that George felt that the foundation has been laid. I mean, we are not in kindergarten anymore, and in fact, nanotoxicology is now more in an adolescent stage. And I think that was sort of the message, that the foundation has been laid during the last 10 to 15 years. And furthermore, not that we should abandon nanotoxicology. Instead, I think we can apply nanotoxicology and nanosafety research as a template or as a general approach when we look at other new classes of materials. It's popular nowadays to talk about advanced materials, but we also just discussed nano and microplastics, which might be considered as a separate group of materials. George was discussing also the information technology and so on, but I think there's another area which is synthetic biology, which is I think probably on the horizon. And what I mean is that we can learn from this the way that nanosafety research has been conducted, which at least in Europe has been conducted in a very collaborative manner, but also in Europe and elsewhere in an interdisciplinary manner. I think this is one of the most important lessons, that we need to collaborate with chemists and physicists and biologists and medical doctors and so on. And of course, environmental experts and so forth. What are the gaps? What is missing? Well, I did highlight something that I think we have learned, and that is the importance of characterization. But in many projects, at least, perhaps it's not really true at today, but in the last, for many years at least, it was certainly the case that we seem to focus all our efforts on very few materials. And so there is a tremendous amount of research on titanium dioxide and silver nanoparticles and carbon nanotubes, and maybe zinc oxide. And that's it makes sense, because these are materials, and silicon dioxide, by the way, amorphous silica. These are materials that are produced in large amounts, and so it's reasonable and sensible to study these materials. But I think we are still, let's say, a little bit far away from fulfilling the promise of predictive toxicology or predictive nanotoxicology. We may discuss later on, I suppose, in the training school, about grouping and categorization efforts. And this is very important, but I mean, how do we really accomplish this? And I think that's one of the gaps that we need to address. How do we move from the study of a few nanomaterials to really predicting the toxicology of the many thousands of new nanomaterials?
Yeah, exactly. I fully agree. And also the toxicity of mixtures or multi-component nanomaterials. I think there is still also more research needed and many things and challenges also ahead.
That's
Okay.
a very good point. The mixture exposure, the toxicology of mixtures and in general nanoparticles or nano and microplastics and other chemicals or pollutants. Absolutely, that is. I think you are quite right. That's another important topic. Yeah.
And I also think that you made a very valid comment regarding this interdisciplinary collaboration. I'm sure you also learned from the trends that now the safe by design concept moves towards safe and sustainable by design. And especially if you think about sustainability, you also include social aspects, ethics, economic aspects. And I think that especially also this interdisciplinary collaboration will also be a hot topic.
Definitely. I mean, it's absolutely true that at least in Europe, safe and sustainable safety and sustainability and safe and sustainable by design are sort of key concepts. Maybe not necessarily new concepts, but there's certainly a renewed focus on this topic, especially in Europe with the Green Deal initiative and so on. Yes, I think that we can. But I suppose this will be discussed also in the training school. Is that right, Susanne? Yeah.
Yes, exactly.
So I don't have to expose my ignorance. I can let other experts speak on this topic. But yes, I think that you are right that this also brings other experts into the discussion, and it's a. I still think that toxicology is toxicology, but you are right that one needs to put the toxicology in a broader societal perspective. Yeah.
Perfect. And let me quickly check if we got a new question. I don't think so. Maybe
People
as a
are
last.
probably getting ready for football at this point.
Yes, true. Actually, Austria is playing soon, so I also have to check how they will perform. Maybe one very last question from my side, since this training school is also focused on PhD students and early career researchers. From your perspective, do you think we have enough young researchers also focusing in toxicology? Or should it be more? Do we have enough next generation researchers that are engaged in this topic and are interested?
I mean, I think there are really a lot of very excellent young researchers in this field, and actually our department at Karolinska Institute offers a master program in toxicology, and it is the oldest master program in Europe. So we certainly train toxicologists all the time, and they learn also a little bit about nanotoxicology, of course. I think that I'm not, let's say, worried. At least from my perspective, I think there are a lot of both students and postdocs who are interested in this topic. I think that. Well, just one comment maybe to add to this is that what is the ideal background? One may ask. I mean, my background actually is in medicine, but many of my students and postdocs, I mean, let's put it this way: my students and postdocs have very differing backgrounds. Some come from biology or molecular biology or biotechnology backgrounds. Some come from more of a chemistry background. One way or another, one has to broaden one's perspective. If you are a biologist, then you have to not be afraid to learn a little bit of chemistry. And if you are a chemist, then you have to make sure that you understand a bit of biology. So this is the challenge for our students and postdocs. But I'm not too concerned, because I think there is a good number of people who are interested in this field.
Perfect. Great. So I think since there are no further questions, Stella thanked you back also in the discussion forum for an excellent talk. So yes, if it's fine for you, I think we can close this session just around 5 minutes before time. And thank you very, very much once again, Bengt. It was highly appreciated that you were available for this interesting. And yes, to all participants, thank you very much for joining today's first day of the training school. And I hope to see you tomorrow again at 1 Central European Time. Have a good evening and bye to everyone.